This is a one muscle fiber. You take your biceps brachii, or first lab you were dissecting the, let's say, trapezius. You pulled out one muscle fiber. That one muscle fiber is going to look like this. I'm going to ask you a lot of short answer questions. If you can answer those short answer questions, that means you got the concept. That means you got all the material that you need. So one big muscle fiber, right? Obviously, this is within the whole muscle. Within the whole muscle, you have fascicles. And within fascicles, you have individual muscle fibers. Inside the individual muscle fibers, you have another cylindrical structure. Actually, I'm not going to go, technically it goes all the way out there, but I'm not going to do that because it's going to be a little bit confusing. This is called what? Whoops.
Sorry.
What is this called? Come on, help me. I forgot. Heidi, what's it called? Gonna help me or what? All right. What is it called? No, we're not there yet. Myofibril. Yes. Myofibril. This is like spaghetti packet. When you pull the spaghetti package out, you're going to see those little noodles come out, one individual fiber. These are called a myofibril. Inside the myofibril, you have two important proteins. One is called the actin. I'm just going to do one of them. Big fat one. Sorry, this is called myosin. One is called the myosin, this big fat one. And the other two, there are two big ones out here. They're thin, like this. We're going to call this actin. Okay? So we're just putting the big picture together, but slowly. So there's a myosin. Myosin, it has these little golf club-like structures. We call them myosin heads. So think about this. Inside of the biceps brachii, when you're trying to do 50 pounds, you're going like this. Let's say, hypothetically, I have 1,000 fibers here. I have 1,000 of these guys. Inside of each of these fibers, I have hundreds of myofibrils, right? So when my brain says, okay, I want you to do this. This is a 50 pound I have in my hand. My brain says, do this. At the microscopic level, this is what's happening. These two green lines that you see here, called actin proteins, they come together. So if you have a lot of sarcomeres, it's a lot of units that they come together like that. Imagine in my arm, my whole arm is going to be shortened and muscle is going to be bulged. So that's what you see when you're flexing your arm. That's what basically doing. You're having all these coming together. Does that make sense? All the actins coming together. Imagine I have 500 of these lined up like this, 500 of these green ones lined up like this. So when my muscle goes this way, and they all come together. We call this sliding filament mechanism. This process is called the sliding filament mechanism. Sliding filament referring to the sliding of these actin molecules over one another. Now, what causes that to move in that direction? Let's add a couple more components. First of all, we know the brain has to tell my muscle to move. So write it down as a general rule so you know every single muscle fiber. I'm not talking about muscle, the whole muscle. I'm talking about every single muscle fiber has to have connection with the motor nerve or motor neuron, like this. This is a motor neuron. If this is cut, that muscle fiber is paralyzed. Simple. So let's go back to what story I was telling you about. If I have 1,000 of these in my biceps brachii, if one of them doesn't have this connection, that one is not going to work. Simple. So that means you have to have 1,000 connection with the neurons. So the neurons, they come down, they branch. Every single neuron is going to be making contact with this guy. Are we good? So this neuron here, when my brain decides that, okay, I got the weight, my brain says, go. This neuron has to do something. What does it do? Releases what? Neurotransmitter. You've learned that in kindergarten. Neurotransmitter is where the neurons communicate, right? What would be the neurotransmitter here? What's the name of it? You've heard of ACH or acetylcholine. That's the neurotransmitter that is released at that junction. This is a chemical. When it's released from there, it comes and binds to these receptors at this junction. These are the receptors. This junction is called the neuromuscular junction. Neuromuscular junction. This junction here. Are we good? Neuromuscular junction. This is where the neuron comes in contact or synapse with the muscle. This jaggedy line that I have drawn here, it's called the motor end plate. I'm just giving you all the microscopic structures. This is called the motor end plate. This one. Motor end plate. If I ask you what's the significance of motor end plate, Brianna, what do you tell me? You say it's easy because the receptors are located there. If I say, Amy, what's the function of the receptors? To receive the signal from the neuron. So there are diseases that actually your immune system destroys these receptors. Guess what happens? Your muscle is paralyzed. You can die from it. So this area is critical. Neuromuscular junction. This is called the motor end plate. If I ask you, what is the motor end plate part of? You say cell membrane. Do you see this? This is the cell membrane of the muscle. By the way, we call it fiber. Why fiber? Why do we call it fiber? Not a muscle cell. Evan? No, because they're long. Yeah, because they're long. So you can actually call a cell if you want to, but we call them muscle fiber just because they're long. Some of them could be up as long as 3 feet. Just like all the way up there, stretches. So that's neuromuscular junction, receptors, motor end plate. The cell membrane of a muscle fiber is called what? Sarcolemma. Sarcolemma is the name given to the membrane, cell membrane of a muscle. It's a special name. It's kind of like the cytoplasm of a cell. The material inside the cell is called cytoplasm. In a muscle, we call them sarcoplasm. So just names. The term myo, sarco, referring to the muscle. So far, so good. Now let's quickly go back and talk about the story again. Brain says, okay, move your arm, right? I'm at this point. My arm is not moving yet. Brain releases, neuron releases neurotransmitter. Neurotransmitter picked up by the receptors on the muscle. This is going to be creating electrical activity down along the membrane, which you're going to learn in physiology, by the way. Here, nothing. You're good. If you just tell me that electrical activity is generated, you're good. By the way, it's called action potential, in case if you want to know. The electrical activity is called action potential. which you learn in detail in physiology class. What does it do? How it gets there, and what channels at the molecular level, what it does to create this, generate this, the electrical activity. This travels all the way along the membrane, which is called what? Muscle membrane, which is called the blank. Sarcolemma. Down deep into the muscle fiber via this structure. This structure is called T-tubule. T stands for transverse. T-tubule, or transverse tubules. So the electrical activity that generated there travels along the membrane and enters the cell through T-tubule. Notice what happens. Inside the muscle fiber, next to the T-tubules, there are these little boxes on either side of it. What do you think these boxes hold?
Calcium.
Calcium molecules. Lots of calcium here. What do you think the function of calcium is? Next time when your doctor prescribes calcium channel blockers, why are they trying to do that? Think about calcium, heart contraction. Heart is a muscle, cardiac muscle. So that means it has something to do with the contraction, and they're trying to manipulate that. Now, this is skeletal muscle. Skeletal muscle and the cardiac muscles are very similar in terms of structure and what they do. There are some major differences, but in general, they're like muscle fibers, muscle tissue, and they do the same thing. So calcium stored here in this little box. Box has a name. Box is called the terminal cisterna. This one here. Terminal cisterna. So that's the name given to these little sacs here, which this terminal cisterna is part of bigger, elaborate network of sarcoplasmic reticulum. Probably you've heard of in your cell biology class. So it's basically these little lines, expands everywhere. I don't want to draw it there. This is called the SR, sarcoplasmic reticulum, which basically, then again, in physiology, you'll learn in detail what is the function of that. Don't worry about it. Just know that it's there. So calcium stored here. Now, once the electrical signals come down here, this electrical signal is going to open up the gates. What gates? Calcium channel, calcium gates. So allows the calcium to be released. Notice that it released inside the muscle fiber. Why is calcium released? For those of you who are sleeping or taking a nap, why do you think the calcium is released? To do what? Remember, my brain said that to move this 500 pounds, and I haven't started yet. I'm still in the process, right? It's exactly. Without the calcium, there's no contraction. So calcium is going to do something, and we already know contraction means basically these two green lines coming together. At this point, there's no way those green lines are going to come together. What's going to move them?
The calcium.
Calcium is going to come in and help to move them. But what does the calcium do? Once the calcium is released from there, exactly, it's going to come over here, does something here to allow those two actin molecules that come together, right? Pardon? Yes, so actually no, here on the actin, we're close, we're almost done. On the actin, you have these little dots. It's called the binding site. These dots, when the heads bind to these dots, the green are going to come together. The actins are going to come together, kind of like a magnet.
Okay.
But at this point, they're not binding. You see that? They're separated. What separates them? Another protein. There's another protein that actually blocks these heads like this. This is called tropomyosin. This protein is called tropomyosin. Tropomyosin is a protein that blocks the binding sites on the actin. So far so good. Bear with me, we got 2 more structures and we're done. Trust me. So those are the binding sites, but these binding sites are basically these heads that you see here. Imagine like these. This is the head, this is the actin. The heads are like magnet. They want to bind, but they can't. Why? Because this guy's blocking. So the function of calcium is to remove that. When the calcium is released, calcium is going to come here and then bind. There's a complex, it's called the tropomyosin-troponin complex. So we have actually another protein. Let's do this in, I don't know. I ran out of colors. Let's do purple. Another protein here on the tropomyosin right here, the purple one, like this. This is called troponin. Have you heard that when somebody has a heart attack, they go and they measure their troponin level in the blood? Same thing. It's a protein. If it's damaged, it's a sign of that individual had a heart attack. Actually, when we get to cardiovascular system, we'll talk about this a little more. So, this protein, troponin, it's there, sitting there. For what? Acting like a handle, door handle. So, when calcium is released, it comes and binds to troponin. Once it binds to troponin, it pulls the tropomyosin off of these binding sites. Once they're pulled off, the head's like magnet, they're waiting. They jump up, grab onto these binding sites, and then move the green lines forward against one another. We say, congratulations, the muscle contracted. Piece of cake, right? Now, question, the muscle did this, right? I did the contraction. Then what? I have to relax, right? What am I supposed to do?
The calcium goes away.
That's where the trick is. You need ATP. That's where the energy comes in. That's what you're guessing, right? Yes.
Yeah, you learn it in everything.
So that's where the energy comes in, and that's why you need to have 15 hamburgers to be able to survive in this class for dinner. Because yes, you need ATP. You need to generate a lot of ATP, my friend. So that's not a lot, by the way. 15 is not a lot. So ATP is the one that comes actually stops this process. As a result, the heads go back. You can relax. So you've heard of rigor mortis. After death, you're not producing ATP. And guess what? Muscles get contracted, the body gets stiffened. So you get a lot of calcium in there, the body is very stiff. And then after several hours, after bacteria start partying there, then those things fall apart and the muscle loosens. But otherwise, the muscle is stiffened because you don't have any ATP coming and releasing the heads. So this is the story. If you can take all these components, write it down, starting from the axon of a motor neuron, acetylcholine or ACH, neurotransmitter, motor end plate, receptors on the motor end plate, sarcolemma, action potential, release of calcium, or actually T-tubules, release of calcium from terminal cisterna, binding of calcium to troponin, removing the shape of tropomyosin, binding of myosin heads, and contraction of muscle, and ATP releasing the muscle heads. That is the microscopic anatomy of muscle. We covered the whole lecture in 20 minutes. Can't be that hard. Right?
Right, sure.
There we go. All right. I think we stop here. So this will give you a little thing when you get a chance. Clean this up, draw your own diagrams, add some information. By the way, in your lecture notes, I have a page that actually describes these things in one page. So if you want to, work it, look at it.